Okay, this is a voice recording for your muscles unit. We're going to start off with the organization of the muscle, and how it's connected to the bone with the tendon here. Break it all the way down to the main motor unit, known as the sarcomere, and then we'll look at some of the terms associated with this unit, and then we'll start trying to identify the major superficial muscles that you have to know for your test. So let's start off. Here's a muscle, as you can see, attached to the bone. Remember that tender tendons, right? Muscles tend to be tender. Tendons attach muscle to bone, whereas ligament is going to be bone to bone. In this picture, you can see that we have the major muscle. Maybe this is a bicep brachii or tricep brachii, but we have a muscle, and the muscle is made up of several smaller subunits. Around the muscle is the epimysium. Remember that prefix, mysium. Mys means muscle, epi means surface. So this is the material that wraps the muscle. And then you have these extra bundles inside those, and those are the fasciculus. Okay? And inside each one of those, we have the muscle fibers. We termed these myofibrils. And inside those myofibrils, we have extra little pieces within those. And those extra pieces inside those myofibrils are the myofilaments. So inside here, the myofilaments. And know that there are two myofilaments that we have to know. One is actin, and one is myosin. We'll talk about those in a little bit. Actin is your thin, myosin is your thick. We'll identify the different pieces and how those things allow for the contraction of your muscles. You can also see some of the other terms here that might be on the test. Endomyosin, right? Endo means inside, so we're seeing inside these bundles is the tissue that supports them, and then between them, hence the peri, is the tissue that helps support those bundles, so that there's not really any open air mass whatsoever. There's liquids and other tissue materials that help hold all these muscle pieces together so that they all work in one central unit. Keep in mind that muscle is a multinucleated piece, and that your fibers will end up getting thicker, but you really never add extra fibers as you develop muscle tissue over the time period of your life. All right, so here's one of our main pictures that will definitely be on your test. Not exactly the same one, but a very similar one where you have to know the different pieces of just one of these fibers, right? So this is a myofibril, and you can actually see the myofilaments, the little dots here, and you have one that we labeled out on your diagram packet that is very similar to this one, where the thick dots you have labeled as myosin and the thin ones you have labeled as actin. The reason that we need to really look at this is each one of these little fibers has a wrapping called a sarcolemma. Right, so this is the wrapping that supports that fiber, and that sarcolemma will kind of contain and house all the nutrients that we want to separate from maybe the other fibers, because we might not want to recruit all the fibers depending on the type of reaction that our body is trying to elicit. So as we'll talk about the neuromuscular junction, neuron connects into this and allows for things to pass information. The T-tubule is going to be one of the things that helps first distribute some of the calcium that's going to allow for the muscle contraction. That calcium will travel down the T-tubule, and then it will start moving through the sarcoplasmic reticulum, which is, in your diagram, it looks very spiderweb-like across the surface of the myofiber, and that will help distribute the calcium in a more even distribution all along. You can kind of see that there's sarcoplasmic reticulum in between these fibers as well, allowing for the maximum surface area and distribution of that calcium. Also keep in mind that muscle contraction largely an energetic process and takes quite a bit of the molecule ATP, and your organelle that you should definitely know by now that makes all this ATP, or majority of your ATP, is the mitochondria. So this muscle tissue has lots and lots of mitochondria. So if you're looking at a muscle tissue that had a lot of effort putting forth in a long period of time, something like a dark tissue or a slow twitch fiber, not only is there going to be a lot of myoglobin, but there's also going to be a lot of mitochondria in those tissues to allow for a long sustained ATP production. The other pieces that are in this diagram that are of note, I think I already talked about the T-tubule, the sarcoplasmic reticulum, the sarcolemma, right, and then those actin and myosin fibers, which we're going to look at here. So you can see it's already labeled thin myofilament and then thick myofilament adenomyosin. So let's look at this. So here's showing you a microscopic picture of the basic drawing that you're seeing here. So between Z lines is called a sarcomere, and those are your working units of the muscle. You can see that there's some darker lines in here and some thinner lines as well. The thin ones are your actin, so thin filaments here, actin, and the thick filaments, the myosin, which be wedged in the center. The two of these units will work together, and you'll see in another image here that the myosin has these little heads. It grabs onto the actin and it slides the actin molecules together. So this H zone, as you're seeing here right now, is in a relaxed position because there's space in the H zone. Eventually, if we get a contraction, those myosin heads pull, the actin fibers will move towards each other in a contraction, and the H zone pretty much goes to disappearing. The Z line will then get closer and closer together, so the distance between Z lines gets smaller as the muscle contracts and longer as the muscle relaxes. So make sure that you understand what the Z line is, what the H zone is, myosin, actin. I don't believe we have I band on the test, but it's of note that the I band is the actin distances that we're looking at here, and that how it transverses one sarcomere and goes into the other sarcomere as well. The next image is showing the neuromuscular junction, which you do have to know because we're having this, our next unit is going to be on the nervous system. We have neurons motor neurons that interact with muscle tissue. So that's what you're seeing here, that we might have an action potential, and if you don't know that, we'll talk more about it now, where we have this electrical jump coming down the neuron, and eventually gets down here at the end of the axon, and instead of sending an electrical impulse straight across this gap, they generally release chemicals, neurotransmitters. The main one that you have to know is acetylcholine, or ACH, which allows for a postsynaptic. So here's postsynaptic, here's presynaptic, because the space between is the synapse, right? So there's no physical contact here, it's a space. That ACH will allow for the postsynaptic neuron to get the change. Usually it's gated change, so there's little surface molecules here, usually carbohydrates, they're sticking up, and proteins that are sticking up on the other side of the cleft, and they will respond to ACH attaching, and then molecules will rush in, generally sodium, and cause the postsynaptic cell to do a change in its composition, in the way that it's working, and in turn we'll talk about how it actually causes a release of calcium inside this cell to go down those T-tubules across the sarcoplasmic reticulum, and will allow some of the fibers that are covering actin to be exposed and allow for myosin to grab onto them. So key thing here is that you understand neurons are releasing the ACH, causing the muscle cell to react because of it. Here you can see that sarcolemma, that wrapping around that muscle fiber. That's going to cause that sarcolemma to get the signal. Keep in mind that there's an enzyme that is also in your body that will break down ACH, because we don't want constant contraction in the muscle, and that's acetylcholine, or ACE. ACE, or ACE means that it's an enzyme. We'll talk about a disease where the body acetylcholinease gets broken, and in turn your muscle contracts and never really relaxes. Here's the diagram that I was talking about with the binding sites. So when calcium comes through and gets released across your actin and myosin, actin has tropomyosin, which is this threaded material that I believe you have a diagram in your diagram packet for. You can see that the darker circles are covered up right now by this fiber. When calcium binds in, which is troponin that it's going to bind to, it causes the fiber to slide off of the attachment sites on actin. And since those attachment sites are now open, if we had a myosin fiber here, the myosin fiber can now grab the actin and do the work that we want the muscle to do. So if there's not calcium being distributed, there's no muscle contraction. Okay? So here is showing you that actin-myosin interaction. We can see that H zone here. We see the one half of the I zone as well, or an I area. So we have the sarcomere, we have the myosin in the center, and the actin along the wedged in between each one of those. And we're going to get a movement towards each other, and so the H zone will shrink. And the way it does it here on the smaller level is the myosin will grab onto the actin only if the tropomyosin slides off and it exposes those attachment sites in the actin. Here it's the grab and the pull and then the release. What's not shown here is that ATP is required for that release. So if ATP is not available to allow for the release, the muscles actually stay in a fixed position, which we'll talk about that symptom as well. So ATP required for this process. It's just not showing here. You have to understand how this cross bridging and this linking and power stroke works. All right, terms that you have to know: muscle atrophy and hypertrophy. Atrophy is the breakdown or the shrinking of the muscles due to a lack of use. Hypertrophy is the exact opposite, where the muscles enlarge because of use. White and dark muscle. We're just talking about fast twitch here with white and slow twitch here with dark. Generally see lots of myoglobin in the dark or slow twitch fibers and less myoglobin here in the white. So if you're talking about turkey meat, the legs that the turkey is walking on constantly tend to be the dark meat where there's lots of myoglobin, and the white meat or the power stroke meat where there's lots of power in a burst of energy tends to be the breast meat of the turkey. Origin and insertion you have to know as far as muscles are concerned as well. Origin is the fixed location of the muscle attachment. So if you're talking about the bicep brachii, that fixed position is going to be up near where the clavicle and scapula are, at the glenoid cavity area. And insertion is where the movable location of the muscle is going to be. So it would be down near your ulna and radius to allow for that contraction of bicep to move your hand towards your face. Fatigue and cramp are things that you need to be aware of as well. Fatigue and cramp. Cramp is when the muscles go into a fixed, overused situation. Generally, this can be because of dehydration or minerals. Fatigue is after the muscle has been used a long period of time, it tends to get fatigued. Isotonic versus isometric. Isotonic is a movement or a muscle movement where you're doing it all in one motion. So something like maybe if you're doing weights in the gym, you're doing a bicep curl, that would be isotonic. Versus isometric, this is where it's a fixed set of muscle use, but you're not moving anything. So those of you doing wall sits, where you sit in a seating fashion position, but you're not actually in a chair, that would be an isometric type of activity. Both of these are very much parts of training regimens for athletes out there, and they're both equally valid things to develop muscle and balance. Tetanus and rigor mortis are things that show up on the test that you have to know about. Tetanus is a syndrome caused by a bacteria where acetylcholinesterase that we just talked about gets damaged, and it can no longer break down your ACH, and in turn the muscles go into full contraction and never get released, and this is a permanent syndrome. So if you develop tetanus, that's pretty much it for you, unfortunately. Rigor mortis, just like it's saying, rigor means muscles going into clenched state, mortis means death. This happens when the body is not making any more ATP. The muscles go into a fixed state and will stay that way for 12, 24 hours, depending on conditions and whatnot the body is in. And eventually they'll relax because the body starts decomposing based on bacteria, so that muscle will eventually become more flaccid. All right, let's talk through some of the major muscles that you have to know. Pectoralis major, you definitely have to know. Deltoid, you definitely have to know. You can see these separations very nicely in this model. Sternocleidoid and mastoid, you have to know. The ones on the face that you have to know, orbicular oculi, you have to know. You have to know the masseter. It's the one that allows for the jaw to close. Frontalis, the ones up here in the front of the head. And then temporalis, the ones on the side. You have to know your rectus abdominalis and how that all comes all the way down to the pubic bone. Serratus anterior and external oblique. These are the two on the sides here. Latissimus dorsi, you can kind of see in this image, just kind of wrapping around. It's usually easier to see the latissimus dorsi in the back of the individual. You have to have, this one says coracobrachial. You just have to know this is bicep brachii. And you have to know on the arms here, definitely brachioradialis. Right? You have to know some of the other smaller ones that are in the arm as well, which we'll talk about in a little bit here. You do have to know some of the hip flexor muscles, like the pectinis, which is right here. You have to know the iliopsoas, which is just above that. You have to know the gracilis, right, which is on the inside of the leg, and then the adductor longus, which is this one that's kind of wedged in between those two. Sartorius, the one that kind of wraps around and goes to the inside of your knee. Rectus formalis, one of the ones that is considered part of your quad muscle. You have to know vastus medialis. Medial means midline, so here's medialis. And vastus longus. On the lower leg, you have to know the tibialis anterior, right here. Right, helps raise your toes. And the peroneus longus, just outside that. On the back of the leg is the gastrocnemius, which is kind of hard to see in this diagram, so we'll flip and start looking at another diagram. This one's a nice one as well. Shows a lot of those muscles. Some of the ones we've skipped are in these windows, and you kind of notice that as you were working with your models, putting together those clay models, that we didn't really talk about serratus, or not serratus anterior, but some of the ones that are up in this area, like teres minor and teres major, and infraspinous. You don't have to know those, but you do have to know trapezius. It goes all the way up the back here, right? Here's the latissimus dorsi, noticing it's coming all the way down to the top of your pelvis. There's the ilium of your pelvis. Gluteus maximus. You don't have to know medius, but gluteus maximus for sure. Bicep femoralis, you definitely have to know. You do not have to know semitendinous, even though it's right there. I'll just kind of left that one off because it's a little harder to see in some of the other diagrams that you have. Gastrocnemius, and then the soleus, kind of along the sides of the gastrocnemius, are ones that you definitely have to know as well. As far as muscles, I think that covers the majority of them. If there are other ones that I've missed, by all means, we can discuss them. If you missed some of them, I'd be more than happy to help you out. Otherwise, most of the diagrams that you have in your packet will be on the test. There will probably be a couple extras that are very similar, but for the most part, pay attention to that practice quiz. That'll help you out a lot. I wish you the best of luck, and see you next time.